we will be discussing about the pyrosequencing. The word itself says that it is a sequencing method, and in the pyrosequencing, the sequencing is done by the detection of the light. So, what is the sequencing? Suppose we are having a DNA and we do not know what is the sequence the nucleotides are being arranged. So by using this type of sequencing, there are various types of sequencing like Sanger sequencing, pyrosequencing, Maxim-Gilbert sequencing. In this video, we will deeply discuss how this pyrosequencing is done. So let us see what are the components which are required for this sequencing to be done. The very first thing is the DNA template to whom we need to be sequenced. The other thing is the DNA polymerase. The work of this DNA polymerase is that it will add the nucleotides or the DNTPs. The next thing is DNTPs, which are ATGC, the nucleotides which needs to be detected. The next thing is the ATP sulfurylase. It is one of the substrate which is used in the pyrosequencing. The next is the luciferase, luciferin, epirase, APase, adenosine 5-phosphosulfate. We will discuss the function of all this in this video. So, there are various types of sequencing methods. So, why we are using this sequencing method? This sequencing method can be used when the base pairs are less. So, it has advantage as well as disadvantage. If we are having a short fragment of this DNA, we can easily detect this or we can easily sequence it. But what if there is base pair near about 1000 base pair? So in that case, what we have to do is we have to cleave the DNA into fragments, then only we can do the sequencing. In this pyrosequencing, we can detect from 300 to 500 nucleotide base pair. But in case of Sanger sequencing, we can sequence it from 1000 base pair. Up to 1000 base pair, we can sequence it. So, if we are doing the sequencing, where it is used, this can be used for the detection of single nucleotide polymorphism. If in the DNA there is some changes in the insertion or deletion of the nucleotide for which the whole in the genetic level, the whole mechanism is changed. By this type of sequencing, we can detect it where the nucleotide is being changed. Or we can quantify the DNA methylation as well as allele frequency. If there is a DNA and in this DNA, in some places there is a methylation. This methylation can be detected by doing the sequencing. In any kind of sequencing we can detect, but if it is the small nucleotide, we can perform this pyrosequencing. As well as we can use it for seeing whether there is insertion, deletion or other sequence variation in the DNA. So let us see what are the things required for this whole process to be done, how it is done. So the very first thing which is required is a DNA. The DNA which is present, it is in the double-stranded form. So if we don't have a plenty amount of DNA, the first thing which we have to do is we have to do a PCR reaction. By this PCR reaction, we can get plenty amount of or the multiple amount of this DNA fragment. So if once the experiment or the sequencing is failed, we will have some of the DNA in the backup. The other thing which we need to do is we need to denature this DNA because the DNA which is present, it is in the double-stranded form. First of all, it needs to be in the single-stranded form. So for that, we have to denature it. The other thing which is required is a primer. Why do we require a primer? For the synthesis of the DNA, we require a primer. But the DNA which we are taking, suppose we are taking this DNA, then what we have did, we have denatured it. So we got a single-stranded DNA. We don't know the exact sequence or we don't know any sequence of this DNA. So how can we design a primer? So for this, there is a ligation of the adapter and some of the known sequence in the terminal end. So here the pink portion which is present, it is the adapter sequence. This adapter sequence is the known portion. From here we can design the primer according to this adapter. The next thing which is ligated is the biotin. This biotin is ligated because it can bind with the bed. So for the process to be done, the first thing is that we have to prepare a library. For this preparation of this library, there is a magnetic bed in which there is streptavidin coated. This magnetic bead is coated with the streptavidin, and this streptavidin has a binding affinity with this biotin. That is the reason this biotin is being ligated over here, so it can easily bind with it. So here we can see that there is a DNA as well as DNA bead as well as single stranded DNA. So now we got that the single stranded DNA is being attached with the DNA bead. The next thing which we have to perform is emulsifying PCR. This is the product of emulsifying PCR. Here there is a bit. Now the single stranded DNA which was present with the adapter, here it is being amplified, or we are getting the multiple products of the single stranded DNA. So the beads which is present, so the bead which is coated with the streptavidin, it can attach millions of the single stranded DNA. Now these beads will be placed in a reaction plate. In the reaction plate, there are multiple well-like structure, and in this one well, there is only one bead attached. Only one bead can be incorporated into it, and in this one bit there will be multiple single stranded DNA. And for the whole process to be done, these are the enzymes, that is luciferase, APase, luciferase, DNA polymerase, and ATP sulfurylase. These things will be added into the reaction plate in each well. So now let us discuss how this process is taking place. So the very first thing is that what happens when a new DNA is being synthesized. Suppose here what we are having a DNA. This is the DNA. This is a bit, DNA bit, and attached to the DNA bit, there is the single stranded DNA which needs to be sequenced. So what we have to do is we have to set its complementary strand. For this, it requires some of the enzymes. Before the enzyme, it needs a primer. This primer will be complementary to this adapter sequence. Now we know this adapter sequence. The adapter sequence which is being ligated is a known sequence. According to the adapter, we can sequence a primer. This is the primer which is being arranged. Now this primer is having a 3' OH group for incorporation of the new nucleotides. When the new nucleotides are being incorporated, it releases the pyrophosphate. So for this, what is required? There is a involvement of the enzyme which is known as DNA polymerase. The DNA polymerase help in this attachment of the DNTPs. When the new DNTPs are being arranged, are being incorporated, or is being added, it will be ligated in this 3' OH group. When it is being ligated, it releases a pyrophosphate. That means 2-phosphate is being released. This is the first step. Now, what happens to this pyrophosphate? How it is being detected? This is a pyrophosphate which was released. This green is a pyrophosphate. In presence of a substrate, that is APS, that is adenosine 5-phosphosulfate. It is adenosine 5-phosphosulfate. So this APS will be converted into ATP in presence of the enzyme ATP sulfurylase. Here there is the involvement of the enzyme whose name is ATP sulfurylase. What it will form? It will form the ATP. This ATP in presence of one substrate, that is luciferin, and in presence of the enzyme luciferase, will be converted into oxyluciferin and light. So here the ATP which was present, it will be converted into oxyluciferin in presence of the enzyme luciferase, and it will produce this light. When this light is being produced, it will be detected by the detector. According to the intensity of the light, we can say that how many nucleotides are being arranged, what after the other.